Right, then another thing that links to genetic modification and also to things like cloning is the use of stem cells. The first thing we have to know about stem cells is that they are undifferentiated cells. Or you can say unspecialized cells. Okay, and this is the thing that makes these stem cells so important, okay, especially to scientists. Okay, the fact that stem cells are undifferentiated or unspecialized cells mean that we have the potential, or we can, they have the potential to be given a specific job. Okay, so a scientist can actually facilitate this process and take a stem cell and we can use it to make blood cells. Or, as the scientist, you could take stem cells which are undifferentiated and you could kind of in a lab facilitate that they become skin cells. Okay, so epithelial tissue. We are even able to kind of make them into cardiac cells, okay, and cardiac tissue. So stem cells for scientists are very, very important because they are undifferentiated and unspecialized. They have not yet been given a job. So as the scientist working with these stem cells, you are going to be able to initiate and use this in your research because you are able to decide and assign a job to these undifferentiated cells. Okay, there are two main areas or two main kind of ways in which we can harvest stem cells. Okay, and the two most popular, okay, one is probably more popular than the other because the first one might take a really long time. Okay, in the placenta and the umbilical cord, okay, of a baby, you find a really rich supply, okay, of stem cells. Okay, so when the placenta has been delivered and the umbilical cord has been cut and these two are connected to each other, okay, what you are actually able to do nowadays is you can cryogenically freeze stem cells from the placenta and or the umbilical cord, which can be kept either in Cape Town or in Belgium, and you can use it kind of as an insurance policy for your baby. Should something go wrong, your child's stem cells, remember, are 100% DNA matched to your child, which means there's no worry about the cells not being a match, and now you have to find a donor and things like that. So should the child develop something like leukemia, you can actually use these stem cells from that child's placenta or umbilical cord, and then, you know, we can try and treat the problem, which in that case, you know, could be something like leukemia. So in the placenta and the umbilical cord, we find these stem cells. Okay, but then we find another source of stem cells, and that is in the embryos, okay, of organisms. Okay, so embryonic stem cells, okay, is another way in which we can kind of harvest stem cells. This is a very contentious argument, though, okay, and it's a very contentious way in which to harvest stem cells. If you think back again to early in the year, and when you did reproduction, okay, earlier in the year, you know that a haploid sperm cell and a haploid egg cell have got to fuse to give you a diploid zygote. Okay, and then what happened is, through the process of mitosis, that zygote went from being one cell big, okay, if we kind of make this relevant just to humans, you had 23 chromosomes in the egg and 23 chromosomes in the sperm cells. It combined to give you a diploid number of 46. Then that zygote had to grow, it had to get bigger. And that one cell, okay, doubled up the genetic information and it split. And now that organism was 2 cells big. Then that cell and that cell each divided, and eventually the number just carried on exponentially increasing because of mitosis. Now, embryonic stem cells, okay, can be harvested. Here is a nice little picture where you can actually see a stem cell being harvested from an embryo. Okay, embryonic stem cells are harvested from an embryo. Lately, we have gotten to a point where, if you can see over here, we are in the 8-stage or 8-cell stage of this embryo's development. You can take one of those stem cells, okay? As of yet, these cells have not become specialized. They have not been given a job. They have not differentiated yet, okay? You can remove one of those cells and use that stem cell for research, and it will leave the rest of the embryo intact, and you won't have destroyed the embryo.